Hello, my name is Shahan Derkarabadian, and I'm a postdoc at the Museum of Comparative Zoology at Harvard University. So for this talk, I'm going to present what I see as a potential solution to the problem of delimiting cryptic species when you theoretically only have genetic data. Now, this was a part of a project we published earlier this year, and the paper has the exact same title as this presentation. So in species delimitation, we generally want better and better computational tools and more data. However, I feel that this is increasingly to the exclusion of considering the biology and natural history of the organisms we are studying, specifically how their biological characteristics can affect the results of these analyses. So species delimitation can be difficult for many reasons, one of which is that speciation is a continuous process, and it can be difficult to know when and where to draw the line between populations and species. It's also difficult due to the sheer diversity of taxa we are trying to delimit, particularly when we are relying on genetic data only. And this is because different organismal types have different underlying genetic patterns. For example, the intraspecific genetic patterns of the monarch butterfly will be very different relative to, say, a beetle that's only found in a single cave. So how we delimit species with genetic data should be dependent on the biology of the organism. Now, the organism I study are a group of arachnids called Opiliones. Now, they are indeed quite interesting and diverse in many ways. However, for this talk, I'll just have to say that many species of Opiliones have a common set of biological characteristics. So many species of Opiliones undergo non-ecological speciation, and this includes many different aspects. For example, what I call nested allopatry, where different populations will be completely allopatric and different sets of populations will be completely allopatric, all the way up to sets of closely related species where none of those species ever are sympatric. So that nested allopatry is largely due to niche conservatism, and this together often leads to morphological conservatism across species boundaries. Next, many Opiliones show low dispersal ability and high ecological constraints, meaning they have very specific microhabitat preferences. These two together can lead to very high levels of population genetic structure. And all of this together has the potential to lead to cryptic species. And these characteristics in cryptic species are, of course, not only limited to Opiliones or arachnids, but are found across the tree of life. However, it's pretty extreme in Opiliones where we can have situations like this, where there are 24 different species, each known from one or a few localities across a relatively small geographic range, and all of these species are allopatric with each other. So in taxa like this, when we're trying to delimit species, we often see that morphology and ecology will lump species diversity, and we can't rely on reproductive isolation because these taxa are always allopatric. So we are relying primarily on genetic data. However, there's an issue when we're trying to delimit with genetic data in that many of these programs will actually overestimate the number of species because they are in fact delimiting population genetic structure and not species. And this is a common issue that has been demonstrated many times across all sorts of arthropods, also vertebrates and plants. So this is well known. So now the question is, how can we delimit cryptic species using genetic data? And the approach I attempt here is to use supervised machine learning. Now, generally, these are classification tools where we can use a labeled training data set to inform an unknown data set. And I attempted this approach in this species called Theromaster bruneus. So this species is distributed in the southern Appalachian Mountains. You can find them in mesic forests underneath logs and rocks. Now, this species has an abnormally large geographic distribution for a single species of northern temperate Opiliones. That, coupled with the fact that the southern Appalachians are a biodiversity hotspot, makes us wonder about cryptic species within this lineage. So across the southern Appalachians, the majority of taxa of the plant and animal taxa in this region show multiple species across this same geographic range. So Theromaster bruneus is quite unusual. So With this in mind, we conducted an integrative taxonomic assessment of this species. So starting with morphology, here are some general habitus images from across the distribution. I'll point out this single specimen here, of Roan Mountain. I'll be mentioning this one several times. This is the only specimen we found that looks diagnosable from the rest of Theromaster bruneus, which is here, and they are all pretty much morphologically homogeneous. So in addition to kind of general morphology, we looked at specific characters, including sexually dimorphic chelicerae here of the males, which tend to have these protrusions and tubercles sticking out of the chelicerae. Across this character, we saw no fixed variation from across the distribution. Same with pedipalps, there was no fixed variation across the distribution. Most importantly, looking at male genitalia, there was no fixed variation across this distribution. Now, looking at genetic data, first I'll talk about DD-RAD data. So we collected DD-RAD data from all of these populations across the distribution. And I'll point out a few things here. First, that Roan Mountain sample is the most early diverging lineage of Theromaster bruneus. Now, we only have that single specimen for this lineage, and it's a female. We have attempted to find more of this morpho species, but we have failed multiple times. So looking at now this lineage here, which is the morphologically conservative group, we see that there are multiple genetic clades that are geographically cohesive for the most part, and in some cases are delimited by these riverine barriers. So taking a closer look at genetic structure, we see that within this lineage, there are significant amounts of population genetic structure. So next, for a subsample of these taxa, we collected UCE data for species delimitation analyses. Now, looking at the topologies, they're largely congruent, with the exception of this purple clade, which is monophyletic and DD-RAD, was not in UCE data. Otherwise, it's largely congruent. We also collected Co1 data. Now, with the exception of the Roan lineage and this southwest lineage here, the rest of this here was not congruent at all between Co1 and any of the nuclear data. Okay, so kind of summing it up so far, we have deep genetic divergences within Theromaster bruneus, and specifically here, these lineages are all morphologically homogeneous. So the question is, are these cryptic species? So we conducted a series of population genetic clustering and species delimitation analyses. And to make a long story short, there was no agreement across data types or analyses. And I'll specifically mention SNAP, which is commonly used as a species delimitation tool. When we did it with the RAD data, it favored 3 species. Now, I'll mention that even before we did species delimitation analyses, we were thinking that we had a gut feeling of 3 species based on looking at the topology alone. When we did SNAP with UCE data, we see something completely different. It favored each individual as a distinct species. And so what we generally see in data sets like this, in taxa like this, is an increasing likelihood with increasing number of species hypotheses. So in this UCE data example here, there was a peak at 3 species agreeing with RADseq data, but the further we split these species up all the way to the maximum species hypothesis, it eventually surpasses that. The likelihood surpasses the K equals 3. And this is a common pattern. Usually this is quite unusual, but this is a common pattern we see in taxa like this when running these sorts of analyses. So how do we solve this problem and delimit these species with genetic data? So a few years ago, this program came out called CLADES. This is a supervised machine learning approach. It's called a support vector machine learning algorithm. And this algorithm attempts to classify different sequences as either the same or different species. Now, this uses a training data set. And in this case, the training data set are simulated data based on population genetic parameters representing a broad range of plant and animal taxa. So we'll call this the general training data set. They tested this general training data set on multiple empirical data sets and showed that it works, successfully delimited the species that were expected. So what happens when we apply their general training data set to TheraMaster? It delimits every single individual as a distinct species. So why did this fail? And this is how I think about it. Imagine an n-dimensional biological characteristic space. All the biological and ecological characteristics that go into defining a species. Now, some fixed model like the multi-species coalescent or a general training data set may work across a certain space of this n-dimensional biological characteristic space. So, for example, maybe it works in lizards and in butterflies, right? However, I've obviously shown that it doesn't work in these sorts of apiliones. So my taxa fall outside this biological characteristic space where these methods work. So what do? Well, I can make a custom training data set. And that is exactly what we did. So where did I get this custom training data set from? A previous study. A few years ago, we published this paper showing the utility of unsupervised machine learning in species delimitation. Now, aside from the computational aspects of this paper, it was an integrative taxonomic revision of the genus Metanonicus. So, looking at Metanonicus, this is a genus that's found in the Pacific Northwest, and in this lineage, this specific portion of the genus, there were 6 species. Now, you can find Metanonicus in mesic forests, underneath logs, and rocks. So, we collected multiple data types, including mitochondrial, morphology, and lots of UCE data. And with the genetic data, we did many different types of clustering and species delimitation analysis. The important thing is that everything pretty much agreed. We had ended up with 6 highly supported and robust species. So I made this, the UCE data, the custom training data set that we then applied to TheraMaster. So why can we do this? Because Metanonicus and TheraMaster have the same biological and ecological characteristics, the same microhabitat preferences, and the same mode of speciation. Therefore, they should have the same underlying genetic patterns associated with populations and species. So what happens when we applied this custom training data set to TheraMaster? It said 2 species. And I'll note here that we did not include this Roan Mountain sample in these clades analyses because we only had 1 specimen of this. So for the rest of this, it delimited 2. And when we add this 2 to the Roan Mountain, it gives us that 3 species that we found in RADseq, but also that we suspected based on our gut feeling. And it was good to see this result given back to us because, you know, what is the gut feeling of a taxonomist if not knowledge that is derived from years of experience working on similar taxa? So it was very positive to see this, and I feel this is a good way to potentially delimit cryptic species. So in general, this approach is universally applicable. You can make custom training data sets specific to any organism and or data type. For example, I made a custom training data set for low dispersal apiliones and UCE data specifically. So machine learning in general is pretty flexible, and here that flexibility accommodates the biological variability fairly well. And I feel that this approach is a good combination of it kind of combining the objectivity that we desire in computational species delimitation and coupled with our organismal expertise based on years and years of experience. So that's the approach. And with that, I will thank you for your time. And if you have any questions or comments, please feel free to reach out to me. I'd be happy to talk. Thank you.